Hey, welcome to the class on bacteriological water monitoring. So I'm Eric Adnan with the Sno-King Watershed Council, and we're a local all-volunteer nonprofit. We help people protect local creeks and natural areas, and we do that in a couple different ways. We have a community-based water monitoring program that we train people to go out and monitor local creeks. We also have restoration program locally in the Swamp Creek watershed, which is in the Kenmore area. And we also get involved in some advocacy. We're part of Global Water Watch, which is an international organization. And Global Water Watch is the one that developed our training plan. You should have received a manual for bacteriological training. If you haven't, let me know. But we participate with Global Water Watch. They've developed the curriculum and methodology that we're teaching. So our agenda, we're going to talk about what are bacteria and why are they important? What are some of the sources of bacteria in water? And we're going to learn Global Water Watch techniques for bacteriological monitoring. So why we're concerned about bacteria in water is primarily from a recreational contact standpoint. So most of the things that live in the water naturally, like the fish, the insects, other animals that might come in contact with the creek, generally are the bacteria is not that we're concerned about for our human health is not necessarily an issue for them, but it is an issue for our health. So let's just talk generally about bacteria. They're single-celled organisms. You can't see them. They're everywhere throughout the environment, both on our skin as well as inside us. They're responsible for important functions. Essentially, you couldn't live without them. They're also part of kind of the the process by which things transform from one form of matter to another organically, known as the web of life. So they kind of help things move throughout this sequence. So they're super important. But, and there's also many different types of them. So the ones that we're mostly concerned with are called E. coli. Sometimes we also monitor other coliform bacteria, but there are lots of different families of bacteria. And then E. coli, even though we measure E. coli with the technique that we're using, it doesn't necessarily 100% indicate that there's fecal contamination in the water. It is a strong indicator of that and can be correlated with it. But there's one particular strain of E. coli that is actually really indicates that. And you can't, out of all the zillions of different types of E. coli, you can't test for just that one. So it is a good indicator, and that's why we test for it. But there's just one particular strain that's most of concern. So let's talk about sources of bacteria in water. So one of the sources that's pretty common is pet waste. And that's why you'll see this messaging commonly now to clean up after your dog, because in places where people don't do that, and then it rains, that pet waste can run off into a creek, and then that can cause bacteriological contamination. In fact, Kirkland did a really interesting study on the amount of essentially unpicked up dog waste and discovered that that was still an issue. And so they did an experiment with flagging to essentially highlight the issue and see if they could get some behavior change. Another source of bacteria in creeks is failure of sewer systems. So when people connect their home to a sewer, the main sewer that takes the waste to a sewage treatment plant, it connects via a pipe called a side sewer. And in some cases, those can be become old and maybe get affected by earth movement or tree roots or other things, that they can start leaking into the ground. That can make its way into a creek. So that's one potential source. Also, agricultural use, animals that are not fenced out of creeks, also another source. And in some cases, just wildlife. In Bothell, at the UW Bothell, there's a crow rookery, and it's actually a little bit decreased in size from what it was historically. But if you measure the E. coli and in the water upstream of the rookery and downstream, you can find that they're also a source of bacteria in water. In addition to wildlife, if a stream becomes turbid, sediment can be form like a little attachment point for bacteria. So and also another aspect of turbidity and sediment and bacteria is that sometimes bacteria can be resident in the sediments in the bottom of a stream. And so if it gets stirred up due to high flow or some other activity, that can raise the bacteria level. And in fact, one of my water monitors just this past weekend observed this on a creek that he monitors, where they had a high rainfall on Saturday and there hadn't been any rain for a long time, and he found super high levels of bacteria. So we teach a method, just a simple method. What Global Water Watch advocates is do it the same way, do it at the same location, maintain a consistent schedule. If you are consistent in your methodology, then you can generate credible data. And as always, if in doubt, do it again. So if you think you might have made a mistake in your procedures, just redo that test as opposed to take your best guess at the result. A concept in bacteria measurement is something called colony forming units, otherwise referred to as CFUs. And that is a measure of how many bacterial cells are viable enough to form a colony. So this photograph here is of what's called an R card, and we'll talk about those a little bit more in a moment. But a water sample has been put on this card and it's been incubated. And so separate colonies of bacteria have grown. And the way these R cards work is they're color coded. So the blue ones are the E. coli. And so each of those dots is not just like a single bacteria, that's a colony. So it's a colony forming unit. And we end up counting those as how we count bacteria. And that's if you talk to a water quality professional about how they assess bacteria levels, they talk about colony forming units per 100 milliliters. That's kind of the standard. So when people talk about different numbers and CFUs, it's the colony forming units in 100 milliliters. So we do a calculation because we don't collect a 100 milliliter size. We collect anywhere from a 1 to a 3 milliliter size. So based on our sample size, we do some math to extrapolate up to how many that would be if we had a 100 milliliter size. There's different standards for bacteriological monitoring. Global Water Watch has their own set of standards, and they use this kind of a stoplight representation. And so according to their standards, under 200 CFU is safe for frequent contact. 200 to 600 is use caution, and greater than 600 is definitely unsafe. In Washington State, we actually have much stricter standards. And so there's a section of the Washington State law, the Washington Administrative Code, this 173-201A 200, which is the freshwater standards. So this is essentially the legal standard in Washington State for water quality. And it addresses a number of different areas, but one of them is bacteria. And so the standard used to be a little bit more, not quite as strict as this, but essentially the standard now is they want to find it less than 100 CFU and 100 milliliters is considered safe. And to get more detailed, well, let's see. There's a little bit more detailed standard that I can talk about, but essentially, you calculate something called a geometric mean, and you don't want your geometric mean, which is kind of averaging your results over time, to be greater than 100 CFU. And you don't want any single sample to be greater than 320. So if you read that standard, you'll find this is kind of a high level summary of the standard. There's actually even a little bit more detail in exactly what you want to find, but that's the basis of the standard. So there's a concept called listing, 303D list. So if somebody finds that a water body is contaminated and submits quality data to the Department of Ecology, it can get put on this list. And this is the Clean Water Act, Section 303D. And what that means is this is a stream that's documented to have a particular pollutant. In this case, it could be bacteria. So if a stream has been documented and put on this list, then the agency, the jurisdiction that has control over that water body is required to develop a water quality improvement plan. And then in that plan, they want to see a maximum level, which they call a TMDL, total maximum daily level. So essentially, that agency will then do routine testing, maybe once a month or something, and they're looking to make sure that levels are ideally dropping below that TMDL. But if they're not consistently below that, then they have to continue to implement their water quality improvement plan. And then the process of, let's say that conditions get better and you want to get it off the list, you have to go through a delisting process where you essentially prove that the water quality has gotten better. So bacteria is, I'd say, a very important pollutant, which is monitored by cities and counties. So in terms of the steps that we go through for bacteria monitoring, first thing that we do is we get our materials ready. So what we're using is we're using something called R cards, which are made by Roth Bioscience. And this is just essentially like a cardboard card, which has some enzymes on it and a material that's going to cause your water sample to gel. And then you would incubate these cards, and we'll talk about each of the detailed steps. But so step one is we're going to get these R cards. We also want to use sterile pipettes and a thermometer. You'll also need some form of an incubator so that we can incubate our card in our sample after we've collected it. And then we need a data sheet to record our results when we're done with our incubation and counting our colonies. The R cards that we use come in different types. So some of them are E. coli only, which means that the only thing that will show up on that card is E. coli, and they'll show up as blue dots. There's some that also show other coliforms, and this is what we used to use in the past, but we're primarily just concerned with E. coli. So because it's easier to count, and sometimes the colors are not as strictly definitive as that, we found it was just easier for people to count on the E. coli cards. So what we're using right now is the E. coli cards. So the process of sample collection, there's a process that we go through called pipetting, which is where you collect a sample of the sample water in your pipette, and you can do that directly from the stream and place the sample on your card at the stream. You can also collect a sample in a sterile container, and you can put it on the card at another location, because maybe the location at the stream is difficult for you to find a flat spot, or there's other environmental concerns that for why you don't want to do it there. But those are a couple different ways that you can collect your sample. So then when you collect your sample, we use cards that range from 1 to 3 mil capacity. And even with a 3 mil card, you could choose to put either a 1, 2, or up to a 3 milliliter sample on the card. One thing that we found is that sometimes it's difficult for people to get the whole sample to stay on the card when it's a 3 mil sample. So sometimes people choose to go with a slightly smaller, like I typically do a 2 milliliter sample on a 3 ml card just because it's easier to get the whole sample to stay on the card. So you collect your sample, you pipette it onto the card, let it gel for a couple of minutes on a flat surface, and then transport it back and put it in your incubator. So we're going to watch a little video on sample collection. Hi, this is here to talk to you today about how to use the R card for collecting water samples to test for bacteria. So we moved to the R card recently.
Hey, Eric, I can't see anything. It's just a black screen for me.
Oh, is that right? Okay.
I don't know if that's just me, though.
If it's for you, it's probably for everybody.
Same for me.
Okay. Hi, this is Eric Gadget.
That's working now.
Worship Council, and I'm here to talk to you today about how to use the R card for collecting water samples to test for bacteria. So we've moved to the R card recently from the Poliscan Easy Gel technique, and I'll show you the materials that we're going to use and a couple different ways that you can collect and plate your samples. So R cards come in a variety of sizes and types. So we have a couple different types here. We have the R card ECCA, and this is designed for up to a 3 ml sample. So you can see this is a larger card. And then we have the R card ECC, which stands for E. coli and other coliform, in the 1 ml sample size. So you can do a maximum of a 1 milliliter sample with this one. Other materials that come with the R cards are, they come with a sterile container that you can collect samples in if you choose to. And they also come with pipettes. Typically we order the 1 milliliter pipette, but in some cases we like to use the 3 milliliter pipette, although we've discovered it's easiest to actually collect a 2 milliliter sample with this. Now I'm going to demonstrate a few different ways you can collect your sample, and then I'll demonstrate the process of plating onto the R card. So in terms of collecting your sample, there's 3 different ways. One is you can pipette directly from the stream. Another method is that you can use the sample container provided, the sterile sample container, which can be sterilized and reused, so you don't have to toss this after you've used it. And then some programs use a dipper, and so they collect their sample with a dipper, and then they pipette out of the dipper. So I'm going to demonstrate collecting a sample with each of these methods, and then how we plate the sample onto the R card, so you can see how that goes. So with the dipper, this is something that you're going to want to rinse out 2 to 3 times with stream water. Collect your sample. And this is something that some programs like because you can use this without getting into the stream. So I'm going to collect this sample and I'm just gonna set it somewhere stable. You could have a helper who holds it for you, or you can just put it somewhere where it's not gonna tip over so that you can pipette your sample out of that. So then what I like to do is I like to get my pipette ready. So for this purpose, we'll do a 3 milliliter pipette, and we'll do the 3 milliliter R card. Now, if you watch the Roth training videos, they recommend that they either use sterile forceps or sterile gloves for opening this. What we found in practice in the field is that that can be a bit difficult for people. So I think for our purposes, if you just have dry, clean hands and you're careful not to stick your fingers right in the principal area of the R card flap, then you won't have a problem. So I've got my pipette opened. I'm gonna get my pipette out, ready to collect my sample. So I've got my clean pipette, and then I'm gonna take my R card and I'm gonna carefully open it up and just hold the flap back. And now I'm gonna collect a 2 milliliter sample. I could do up to a 3 milliliter sample, and I'm just pipetting directly out of this sample jar here. So I've got a 2 milliliter sample, and I'm just gonna place this on the center of the card and keep it centered. And that's the trick with this technique, is keeping your sample centered. So once I've collected the sample, I'm gonna let the lid drop back, the flap, and you can see how it starts to spread out. And the key at this point is just to hold it approximately level. Luckily, this is a card that's designed for a 3 milliliter sample, and we're just collecting 2 milliliters. So it shouldn't spread too far. It shouldn't spread beyond the edges of the card. The other issue that people encounter sometimes when they're collecting on our card is that if you tip your card sideways at this point, some of your sample can run off. And so at that point, you just have to repeat your sample. So the key is whether you hold it, or some people put it on a clipboard and hold the clipboard level, or you might have a partner help you hold it. You just want to let it sit level until, maybe 30 seconds to a minute have passed. And then essentially it's spread out as far as it's going to spread out. So you can just set it on a level surface, and you're ready to collect your next sample. So I could do up to 3 samples with the same pipette. For our purposes, I'm going to demonstrate collecting a sample with the 1 milliliter pipette. So I'm going to put this one away, and I'm going to use a different sample. The next thing I'm going to demonstrate is collecting the sample with the container, the sterile container that comes with the R card. So in this case, and in every case, remember, if I'm standing in the stream, I want to collect the sample upstream of myself, or I may not even have to get into the stream at all. So in this case, I'm getting into the stream, and I'm collecting the sample in my sterile container. And if I wanted to carry this sample back somewhere, like say to my vehicle or another location, I could put a cap on it, and I could then pipette out of this sample at a different location. So for your site, that may make it easier. In this case, I'm just going to put it on my box here that has my water monitoring equipment. So I mentioned clean, dry hands. So I'm going to dry off my hands so I'm not getting any additional bacteria or contaminants on the card. I could use hand sanitizer if I wanted to be absolutely careful. And now I'm going to get my 1 milliliter pipette out. I'm going to get my R card ready to collect a sample. So I'm just going to carefully pull this back. And I'm going to draw a 1 milliliter sample. And again, I'm just going to carefully place my 1 milliliter sample in the center of the card until the whole sample is there. Let the flap drop back, and then I'm just going to watch that sample and let it gradually spread out. And you can see that if I tip it, it does go in a certain direction. So if I don't hold it level as it spreads out, it's possible it could go beyond the edge of the card. but at a certain point, it just quits spreading out. And you're safe to collect another sample and plate another sample. So for the last sample, what we're going to do is we're going to demonstrate pipetting directly from the creek and then putting it on your R card. So this one I think is fairly safe right now. It doesn't seem to be traveling too much. So I can put this on a flat surface. Just let it finish kind of gelling, which just takes a couple minutes. So now in this case, I could have a partner holding this card, or I could be holding it myself. In this case, I'm going to get it ready to go so that when I collect my sample, I'm ready to put it right onto the R card. So I'm not going to get into the creek. I'm going to just draw my sample, making sure I'm not on the bottom of the creek. Okay, so I've collected my 1 ml sample. Again, I'm just going to pipette, plate it directly in the center of the R card. You can see how it kind of stays together in one bubble. Then release the flap. And again, I'll just wait for it to spread out. And then once it is finished spreading out and appears to be stable, I can set it down. And then we'll be ready to take these to where we're going to incubate them and place them in our incubator. So that's it for now. The next segment, I will show how we incubate the R cards. Okay, anybody have any questions on that section?
No, not for me. Not for me.
Okay. And we will get a chance to practice with R cards on Saturday. Okay, so that was sample collection. And so one thing I didn't mention about sample collection is I showed that we were placing it in one single drop in the middle of the card. One thing that some groups do and some people do to make it so that you get access to all the corners of the card is to use what's called the dots method. So instead of just putting it in one spot, they distribute it in dots throughout the card. So the next thing is putting them in the incubator. So we're going to incubate them at about 35 degrees Celsius. It could be up to 37 degrees Celsius, which is essentially body temperature. Roth says do it for 15 to 24 hours. We recommend doing it for 24 hours. And then you remove it from the incubator and count your colonies. And I've got a little training video to show you guys about how this goes. Everybody see that okay?
Yeah.
Yep.
Okay, now we have brought our sample into the incubator, and I have two different sizes of R cards. So typically we're going to do this, you're only going to use one side, either the 1 ml or the 3 ml size card. I just happen to, for training purposes, use two different sides so we could demonstrate the different ones. But I've got my incubator, which is preheated up to anywhere between 29 to 37 degrees Celsius. With the Coliscan, you needed to incubate for 30 to 48 hours. With the R card, you really only need to incubate about 24 hours. what I've found in practice is you can read the cards after 24 hours, but they're almost easier to read if you take them out after 24 hours and then look at them the next day. The colonies have kind of defined themselves a little bit better, but you can read them after 24 hours. So I'm gonna note on my data sheet my sample size, my incubation time, which I'm gonna start right now, my incubation temperature, I'm going to note that down. This one is 35.5 degrees Celsius, and then my incubation period when it's done. In this case, it says media expiration date, so we would just use the expiration date of our R cards. So all I have to do is open up my incubator. Place the cards in the incubator, and then come back and check on them in 24 hours. Okay, so the next thing after we've incubated it is counting the colonies. So we've incubated it for 24 hours. And I do want to clear up one thing. In the video, I mentioned that it would be incubated between 29 and 37 degrees Celsius. We used to use a different method called Coloscan Easy Gel, and that was the temperature range, but now it's 35 plus or minus 0.5 degrees Celsius. So you pull it out and basically count the blue dots and count the pink ones if you're doing other coliform. And as I mentioned, if it sits outside the incubator for too long, your counts may increase. So I would try to read it within a relatively short time after you've taken it outside the incubator. All right, and I've got a short training video on counting colonies. Okay, so now 24 hours have passed since we first put this in the incubator. So now we're going to open up the incubator and see what we find. And so looking at our cards, what we find is that we do not have any dark blue dots. We just have pink, which means that we did not find any E. coli. Unfortunately, this is kind of a cold and clear period without a lot of bacteria, so it's good from a bacteria standpoint, but not necessarily from an R card training standpoint. However, I do have an example of what we would have found if we did add E. coli. Those would show up as these dark blue dots like we have on this previously incubated card. So these will tend to show up better after an additional 24 hours. You don't gain any additional colonies, but they're even easier to read. So you can take them out now, you can read them now. If you want to make them even easier to read, you can let the card sit around for a little bit longer. So that's how you take the card out and you read it. And then what we would do is we would just count up the number of dark blue dots, record that as our E. coli results, count up the number of pink to purple dots, count that as our other coliform, and that's how we record our result, and we'll just record that on our data sheet. Any questions on that?
No, I'm good.
Okay, so we collected our sample, we plated it on the card, we incubated it, we counted our colonies. And so now we're going to do some calculations of, all right, how many CFUs per 100 milliliters do we have? So we're going to count the number of blue colonies, and then we'll do some math. And so on this card, let's say that we counted up all these colonies. And another question is, what do you count as a colony or what do you not count? So like I can see just a faint dot that you can barely see that's smaller than the size of a period on a printed page. I'm not going to count that. But if it's a distinct blue dot that is easily visible, I'm going to count it. So in this case, we've got 17 colonies. So doing the math, this was a 1 mil sample. So if this was a 100 milliliter sample, this would be 1,700 colony forming units. Uh, depending on the count that you get, if we had counted fewer colonies and we had a bigger sample size, it might come out a little bit differently. But essentially, you base the math on how many colonies you count, what were your sample size, and then scale it up. We have a process of online data entry. And so it will automatically calculate it for you. So really all you'll need to enter is in terms of collecting the data is the number of colonies that you counted and your sample size, and then the calculation will be done for you. So you want to record on your data sheet the number of colonies that you counted. And it's a good idea to have this. So in addition to the colonies, so you're going to record your group. So who are you volunteering with? Who was there? When did you do it? The watershed and water body. What was your site location? You want to measure your air and water. What was your sample volume? Incubation time is what time did you put it in the incubator? Incubation temperature is what was the temperature, and period is generally going to be 24 hours. So this 30 to 48 is a guideline from the old method that we use. So it's going to be 24 hours. And then you're going to record the expiration date of your media. And recording all this data is part of our quality assurance process to make sure that we're actually, for instance, we're not using expired media. So then you'll indicate, did you plate it on site or no, did you do it somewhere else? And then you're going to enter your bacteria count here. So how many colonies you had. And if you are counting other coliform, you would enter that here on the data sheet. You could also, under comments, you can record, what high rainfall or unusually turbid, or it's been a dry period. And then you'll sign your form. And then this is an example of our, if you were using a mobile device for our online data entry with the survey form that we have, you would enter your access code, and then you'd have, we have one of these set up for every site. So this is unique to a particular site where you'd be collecting data. And so you'll just enter it'll give you prompts to enter essentially the same information as on the data sheet. And then I'll show you, this is the dashboard for results. And it looks a little bit complicated just because we're representing a few different things here. So one of the things that we want to track is just what's the average number that we're finding. But the regulatory standard is your geometric mean. And it's a little bit complicated. I'll just say look up average versus geometric mean. Geometric mean is essentially a way of decreasing highs and lows and kind of averaging out over time. And so the regulatory standard wants us to use a geometric mean. So we don't want to find our geometric mean greater than 100. So in this case, you could see for this site, the geometric mean line has been above 100. Here's the 100 line for the standard, but sometimes it's below. So after a certain period of monitoring this particular site, the sample, the geometric mean is within standard. And then the other data point here is the highest single sample. And so this other red line on the back on the dashboard represents highest single sample. The legal standard doesn't want that to be greater than 320 CFU. So you can say that this purple graph has been above. So the highest single sample is what's represented by this purple graph. So when it's down here, it was the highest single sample of that sample was a 0, but sometimes it's bounced up above. So again, what this is telling us overall, the results, the interpretation here is that the bacteria contamination at this site is sometimes outside of what it's supposed to be. So this you can find, we have these for each of our sites. We have these data dashboards now. And so for all the different variables that we're tracking, you can pull this up and then you can zoom in on any of these graphs, and you can also toggle these different variables off and on. And for some of our sites, we also have basically an interpretive site web page set up for the site itself, which gives you a little bit more kind of information about the watershed, the stream, who's doing the water monitoring, and so on. So we could use the dashboard to compare to the standards. And I mentioned we generally want it to be under 100 CFU, and that's the geometric mean. So this is an actual excerpt from the Washington State Administrative Code talking about what the standard should be. So within an averaging period, it must not exceed a geometric mean value of 100 CFU per 100 milliliters, with not more than 10% of all samples or any single sample exceeding 320. So it's a little bit complicated, but that's essentially what we're trying to characterize in our data dashboard. So this is again kind of comparing the dashboard to the standard. So you've gone through that process. Now you're gonna clean up your cards. So basically, once you're done with the R cards, you can essentially dispose of them in regular garbage. If you're working in a laboratory setting or doing a high volume of R cards, you can decontaminate them with isopropyl alcohol before disposal. If your Roth has got some specific guidance for how you dispose of R cards and other materials that might have gotten contaminated. So basically they're saying you can rinse the caps and bottles if you're using those with hot water and then dispose of them or recycle. And you can also use either a microwave, an autoclave, boiling water, an oven-proof bag. There's a whole host of methods that they recommend. Again, I think this is really if you're have a high volume of cards, you're doing this in a laboratory. So far from our program standpoint, we don't believe that these are any more hazardous than other things that go into the waste stream. So you can pretty much just, once they've been incubated, you can dispose of them as household waste. So now you've collected your data, you've cleaned up, you're done with that. And now what are some things that you can do with the information that you collected? Well, there's different ways that you can take action with your data. So you want to think about it, you want to interpret it. What did you find out, and what does it mean? And who do you want to talk to about it? So you could speak to the general public. You could speak to decision makers. You could speak to people who are in charge of water quality for your local jurisdiction. You may want to take samples over a period of time and see kind of what happens. So that might help you decide, like, maybe you want to monitor it for 6 months or a year before you do something with your data, which is probably a good idea. Maybe you want to focus in on an area where you find higher levels of contamination. So you might want to adjust what you're doing. And essentially, you're trying to answer the question, is my water body's water quality getting better or worse, and why? Here's an example of a group of young students in Snohomish. They implemented a water quality monitoring program, and they held a public meeting to highlight their findings. That's an example of a data to action strategy. This is an example from Auburn, where they took a series of simultaneous samples throughout a watershed and highlighted with the red dots where they found high levels versus the green dots were acceptable. In this case, Global Water Watch was working in Africa and Kenya, and they created a watershed map, and they basically laid out the bacteria so people could see visually with the map that they'd drawn and with the samples laid out according to the streams and the watersheds. What did that mean for areas that they're concerned about water quality? Another example of data action, there's some groups that volunteer groups that do collection, water quality bacteria data collection, and it's utilized for a thing called the swim guide. And so that's for like swimming beaches. And that is it for us. So thank you guys for your attention. These are some of the groups that have helped pay for our program. So I'm going to end it here and then see if anybody has any questions.
I have a question, if that's all right.
Sure.
So I'm looking at the King County Watershed Council, or Sno-King County Watershed Council website, and looking at the stream monitoring results page.
Yeah.
There's a lot of ones where there isn't a link. Is that because there isn't data, or why do a lot of these that are listed just not have any links?
Good question. So the hyperlinks are for the ones which are currently being monitored right now.
Mhm.
And the ones that are not hyperlinked are some historic sites that have been monitored. So actually, we're going to get those links up, and thanks for the reminder. But yeah, the hyperlinks are just the ones where people are currently monitoring.
Okay, cool. Thank you.